This is Mounica.
This is Pooja.
And you're watching the Biology Channel.
Today we're going to be talking about meiosis. Yay!
Monica's excited. Honestly, it's pretty ridiculous.
Last week we talked about how amazing mitosis was. This week we'll be talking about meiosis, which is even more exciting. There are two phases in meiosis. In meiosis 1, the homologous chromosomes separate into two haploid daughter nuclei. In meiosis 2, the sister chromatids separate into 4 gametes. This is prophase 1. The homologous chromosomes pair up and crossing over occurs. Also, the spindle fibers form. This step is important because it increases genetic variation in daughter cells. After prophase comes metaphase. Now the pairs of homologous chromosomes line up along the middle of the cell facing each other. The spindle fibers attach to the kinetochore. The next step is anaphase. The homologous pairs separate. They go opposite poles of the cell. Sister chromatids do not separate. The last step of meiosis 1 is telophase. Now the spindle fibers disappear and 2 distinct daughter cells form. Cytokinesis also occurs during this stage. Unlike mitosis, there is little or no interface between meiosis 1 and meiosis 2. Prophase 2 is when chromosomes recondense and the spindle fibers reform, and they attach to the sister chromatids. In metaphase 2, the chromosomes line up along the center with spindle fibers attached to them. In anaphase 2, the chromatids separate into daughter chromosomes, and spindle fibers pull them into opposite poles. In telophase 2, chromosomes finish moving to opposite poles, and 4 haploid cells form as a result of cytokinesis. We are only human, which is why things can go wrong in meiosis. Errors can occur in either meiosis 1 or 2. Non-disjunction is when chromosomes or sister chromatids fail to separate. A non-disjunction in meiosis 1 is when chromosomes fail to separate, and a non-disjunction in meiosis 2 is when sister chromatids fail to separate. An example of a non-disjunction is when after crossing over the chromosomes line up along the equator. Instead of separating, the 2 X chromosomes move towards the same pole. This results in one daughter cell having 0 X chromosomes while the other has 2. Like normally, the cells go through a very short interphase. As the cells continue into meiosis 2, the chromosomes once again line up along the equator. The sister chromatids separate properly and move to opposite ends of the cells. The end result is 4 gametes, 2 with 2 X chromosomes and 2 with 0 X chromosomes. After being fertilized, there are many outcomes due to the number of X chromosomes in the gametes and the chromosome given by the sperm cell. All of the possibilities are listed in this table. One of the diseases that can result from a non-disjunction meiosis is trisomy X. This is when there are 3 X chromosomes instead of the normal 2. Trisomy X is less severe when compared to the other chromosomal disorders. In fact, many people never even find out they have trisomy X. There are rarely ever any noticeable differences in the way people with trisomy X look and act. Some symptoms include being taller than average, epicanthal folds, behavioral and mental problems, and weak muscle tone. Meiosis is very important because of its genetic variability. Independent assortment is one method to increase genetic variability. It is the random assortment of chromosomes. When chromosomes line up at the equator, they line up independent of maternal and paternal sources. Another method is crossing over. This is genetic recombination. When homologous chromosomes touch, enzymes facilitate the exchange of DNA between adjacent chromatids, producing chiasmata. Due to crossing over at random locations, called chiasma, there can be indefinite gene combination possibilities. From independent assortment alone, there is genetic variability. For example, in a cell with a haploid number of 3, there are 8 possible outcomes. This number is derived by doing 2 to the 3rd power. Mitosis and meiosis are both similar and different. They are similar because they are both ways that cells divide and both have the same 5 steps, except meiosis repeats the steps twice. Also, both use spindle fibers to separate. The major differences are listed in the chart below. Meiosis 2 is most similar to mitosis because mitosis is when chromosomes separate into sister chromatids. In meiosis 2, the same exact events happen. The only difference is that in meiosis there are half the number of chromosomes.